Assalamualaikum and hello everyone. Today our group will be presenting topic 11, the influence of DNA concentrations and transformation method on bacterial transformation efficiency. So this study looks at how DNA concentrations and the transformation method affect how efficiently bacteria can take up foreign DNA, which is an important concept in molecular biology and also genetic engineering. Next. So this is the research aim and the biological rationale of our study. The experiment has two main objectives. First, it aims to determine how different DNA concentrations from 10 to 100 nanograms per microliter, together with different transformation methods, influence bacterial transformation efficiency. Second is to identify the optimal combinations that yields the highest transformation efficiency. From a biological perspective, transformation efficiency reflects the ability of bacterial cells to uptake and express foreign DNA, which is essential for successful genetic modification. DNA concentration is important because it determines how much DNA is available for uptake, where low concentrations may limit the DNA entry, while excessively high concentrations may cause cellular stress. In addition, the transformations method plays a significant role in how DNA enters the bacterial cell, as heat shock temporarily increase membrane permeability through temperature change. And for the electroporations, it uses an electric field to create pores in the cell membrane. So by optimize this factor, it can improve the transformations efficiency, reduce cell damage and reagent waste, and enhance the experimental reproducibility. So next. And next for the experiment design, including the control variables and treatment used in our study. The controls serve as a baseline reference where the DNA concentration is fixed at 50 nanograms per microliter, and the transformation method is the applied is heat shock. These control setup reflect the normal laboratory conditions. So the manipulated variables here used in our experiments is the DNA concentration, which is a continuous variable measured in nanograms per microliter, and the transformation method, which is the categorical variable comparing heat shocks and also the electroporations. So to ensure the experiment validity, the bacteria strain will and also the plasmid used were kept on constant so that any observed difference could be attributed. So for the treatment involve varying DNA concentrations at 10, 25, 50, 75 and also 100 nanograms per microliter with each concentration tested using both transformation method. So this means that the design allow us for direct comparisons of how DNA availability and the mechanism of DNA entry affect the bacteria transformation efficiency. So for the next slide, so in this slide, explain about the control conditions and response in our bacterial transformations experiment. So a control environment was maintained to ensure that any changes in result were due to the tested factor. Conditions such as temperature at 37 degrees Celsius incubations and recovery time, recovery medium and volume, antibiotic concentration, shaking speed, and the batch of competent cell and agar plates were kept constant, while unavoidable factors like experimental day or cell batch were treated as blocking variables. So the main response measured was transformation efficiency expressed as CFU per microgram of DNA, which was determined using selective plating and also colony counting, with total colony count per plate recorded as a secondary outcome. So based on our expectation, electroporation should result in higher transformation efficiency, and increasing DNA concentration is expected to improve efficiency up to a saturation point. So next.
Okay, so now I will be talking about why did we choose this design, which is full factorial design. So based on our experimental factors and objective, we have selected a few full factorial design. This is because we have two factors, which is DNA concentration, which is continuous, and transformation method, which is categorical. So a full factorial design allow us to test all combinations of the DNA concentration levels and transformation methods within a single experiment. Okay, next. So why did we choose a full factorial design? This is because this design is well suited to our study for several reasons. First, it accommodates to mixed factor types, which allows us to analyze both continuous and categorical variables simultaneously. And second, it enables direct detection of interaction effects. This is critical because the optimal DNA concentration for heat shock may not be the same as electroporation. A factorial design allows us to test whether the effect of DNA concentration depends on the transformation method. Then finally, the number of experimental runs remains manageable and fits within the practical laboratory constraints, while still providing a biological meaningful understanding of how these factors will influence the transformation efficiency. Okay, next. So now for the replication, randomization, and statistical model, each experimental condition includes three biological replicates defined as independent transformations and technical replication through duplicate plating to improve measurement reliability. So the run order is randomized using a software to minimize systematic bias. And if the experiment must be conducted over multiple days, experimental day can be included as a blocking variable. So for data analysis, we plan to fit a two-way A-novel model, including main effect and an interaction term, represented by the equation as you can see, y equals to mu plus A plus B, and A times B plus error, where A is concentration and B is transformation method. Next.
So for now, we have constructed our DOE in a few softwares in which we used JSP to implement and analyze a manually constructed DOE. And next, we have used Microsoft Excel, and we used it to perform randomization. And the design type that we chose to implement was a full factorial design, and it was defined prior to our data collection and while using DOE principles. Um, Sorry. Okay. So this is our randomized run matrix, which we have done in Excel. And we can see that there are factor settings where both combination of factors are represented onto our Excel sheet, which are DNA concentration and the methods. And then we have used a randomized strategy using the randomized function in Microsoft Excel to create a randomized combination of our experimental runs. So we do this to prevent bias and to also ensure the difference in response is the actual evidence and it was not caused by any other variables. And we have our center point, which is the DNA concentration of 50 nanograms per microliter. And this acts as a center point for both type of methods, which is our electroporation and heat shock. Then this is our design properties. So these are the factors specified in JSP. So we have 5 levels of our first factor, which is our DNA concentration. It is a continuous variable, and we used a scale in which we used 10, 25, 50, 75, and 100 nanograms per microliter. And the transformation method is a categorical variable in which we use heat shock and electroporation. And we use 3 number of replicates. And with our 5 level of the first factor, 2 level factor of transformation method, and 3 number of replicates, we have a 30 total number of runs. And the figure that we see here are the design that we used on the SB.
Okay, moving on to the resolution. The resolution that we use is a full factorial, which means that there's no co-founding of the main effects or interactions, as well as for the biasing we have no aliasing or confounding. And for rotatability is not applicable as this is not an RSM design. And for the data center, for the number of center points, you have 3, which is the data concentration at 50 milligram per microliter. Next. Okay. So for the plan analysis, the statistical model we use is the two-way ANOVA, which is the general linear model, also known as GLM. And then for the equation that we use is shown as shown in the presentation, whereby y represents the transformational efficiency, the CFU per microgram DNA, and then A represents the DNA concentration, B is the transformation method, and for the assumption check, in the JASP we have used the normality of residue, which is the QQ plot and also the Shapiro-Wilk. For the homogeneity of variance, we have tested with the Levine's test, and then for independence is ensured by the randomization. Next. So for the post hoc and visualization, Tukey HSD is used for the transformation method, whereas interaction plots and the interaction plot and also the estimated marginal means is also used as a post hoc and visualization as well. For the decision rules, at a significant level of 5%, the effect size is as shown in the presentation, and for the result reported as LS means around 95% of fluoride, and for the significant interaction is used to interpret simple effects. Next. Okay. So moving on to the quality control, we have used randomization to run randomized order by using Excel, and the plating order is also randomized. Yeah. And we also use blinding, where the plate coded during the colony counting is coded. And then for the quality control measure, we have used the same batch of competent E. coli and also the same plasmid for all treatments, as well as pipetted. The pipettes are also calibrated. We also ensured that negative and positive controls are included. For the risk control, the DNA is less than 100 milligram per microliter to avoid toxicity, and we have ensured the standard electroporation settings, and we also duplicated the plating. And moving on to missing data handling, we have recorded any failed runs, and also we have checked the outliers, and we have ensured that the outliers are not removed without any reason. Moving on. So this is the references, and yeah, this comes to the end of our presentation. I hope you enjoy this presentation. Thank you.